Alright, so the next few slides are more or less going over what we saw in the previous slide in a bit more detail and trying to understand how this all works. So let's firstly consider our insulin. So insulin is the hormone that's produced by the beta cells of the pancreas. So it itself is a short protein composed of 51 amino acids. So the function of glucose is basically to lower the blood glucose level. So that's the key function that we must remember about insulin. So we trigger the release of insulin in response to high blood glucose level. So once we have the high blood glucose level, we release insulin to lower the blood glucose level. So the way insulin actually facilitates this is it will actually bind to protein receptors on the outer surface of the cell, and that will allow us to bring glucose into the cells. And if we're taking glucose into the cells, by definition, you're taking it out of the blood. So this will lower the blood glucose level because it's no longer going to be in the blood. It's now going to be in the cells. But remember, you're not taking all of the glucose out of the blood, just part of it. So because the insulin is now inside the cells, it's going to promote utilization of this glucose. So we can actually use it to do different things. So we talked about some of the different things we can do with our glucose. So we can use the glucose in glycolysis to create our pyruvate, which then feeds into the citric acid cycle and creating ATP eventually, if we go down that track, or we can do the glycogenesis to store the glucose as glycogen. So insulin also has a role in lipid metabolism. So it will actually promote a process where we generate additional fatty acids. So we'll have a look at this in our lipids metabolism lecture. It's called lipogenesis. So lipogenesis is essentially making fatty acids, and we can actually attach glycerol to these fatty acids to store our triacylglycerols. So storage of our fat in the form of triacylglycerol. What about glucagon? So glucagon is also a hormone, and it's also released by the pancreas. But this time it's actually released by the alpha cells of the pancreas. So we saw in the previous slide that the insulin was actually released by the beta cells. So we have the release of glucagon in response to low blood glucose levels. So the reason that we have glucagon released under these circumstances is because glucagon will function to increase the concentration of the blood glucose. So it does this through enhancing or speeding up certain reactions. So these are going to be the reactions that make glucose. So if we do reactions that make glucose, then we can take that glucose from the cell. So they're going to be doing these reactions inside the cell to make glucose, and then we take the glucose out of the cell into the blood. So what is the effect of putting more glucose into the blood? Are we going to increase or decrease our blood glucose level? So by putting more glucose into the blood, we actually increase the level of glucose in the blood. So that's the aim of our glucagon. It's upregulating processes within the cell that are able to produce glucose, which we can take out of the cell and back into the blood. So remember, it's useful to have glucose in the blood at a certain level because we can take that glucose to other locations in the body that require glucose to generate ATP. So we can take that glucose to the brain, for example, that is very reliant on glucose as a fuel. So the specific processes that are upregulated to make more glucose are the glycogenesis, which is the breakdown of glycogen into glucose, and also gluconeogenesis. So we see that both of these processes are being stimulated and they're occurring in the liver. So gluconeogenesis occurs predominantly in the liver, about 90%, and glycogenolysis occurs where we store glycogen, which is in the liver and also the skeletal muscle, except only the liver has the enzyme that's required to convert the glucose into free glucose, which can be taken out into the blood. So that's why both of these processes that are involving increasing the blood glucose level are done by the liver. Okay, so we can see the commonality between these two processes is to make more glucose inside the cell, either from breaking down the store in the case of glycogenolysis, or actually making more from the non-carbohydrate source of pyruvate in the case of gluconeogenesis. So once we have this glucose, we can push it back out into the blood and increase the blood glucose level. So essentially, the glucagon hormone is eliciting the opposite response to the insulin. So glucagon will increase the metabolic reactions that make glucose, so we can take the glucose out of the cell, whereas insulin will actually increase the metabolic reactions that utilize glucose, so it's using glucose either for storage or to generate energy. So the reactions that are activated by glucagon are actually deactivated by insulin, and vice versa. So for example, our glycogenolysis and gluconeogenesis that are activated by glucagon are actually inhibited by insulin. Just like on the previous slide, we saw that we have the utilization of glucose through glycolysis and glycogenesis. So these are activated by insulin, but they will actually be deactivated or inhibited by glucagon. So this is how we can actually make sure that we're not just doing both of these processes at once. So for example, if we were doing the processes that insulin was upregulating and glucagon were upregulating, they'd basically be they'd be fighting each other. So the insulin is utilizing glucose and the glucagon is promoting making glucose. So we don't really need to be doing both of these things at the same time. So we'll do either the insulin set of processes at one time or the glucagon set of processes. And to determine when these are required, we look at the blood glucose level. So if the blood glucose level is high, we need insulin to bring the glucose into the cell, whereas if the blood glucose level is low, then we need glucagon to increase the blood glucose level. So adrenaline is actually released by the adrenal glands in response to things like fear, anger, and excitement. So it's associated with what we call the fight or flight response. So it's giving us fast energy to deal with a looming threat, essentially. So you might also see adrenaline called epinephrine, which is what we call it in the USA. So you might see it in either of these representations. So adrenaline actually has similar functions to glucagon, as it's actually able to break down the glycogen to release our glucose. And we're also going to be making glucose from gluconeogenesis. So both of these processes meaning we're making glucose. So we make this glucose to actually have quick action, so we can quickly use this glucose to make ATP. So this time we're not using the glucose to increase the blood glucose level, we're actually using it to generate ATP quickly. So for example, we have the muscle cells responding to the adrenaline, breaking down the glycogen, and also we have gluconeogenesis occurring, which is happening in the liver, and then we can take that glucose to the location where it's required, for example, the muscles. So essentially what we're doing here is we're getting more glucose that we can quickly turn into ATP to deal with the looming threat when you're faced with a fight or flight type situation. So adrenaline will also have an impact on the lipid metabolism. So it's actually going to stimulate the breakdown of our lipid stores to derive energy from those. So we can use our stored lipids to create ATP as well. Alright, so I've shown this overview of carbohydrate metabolism in quite a few different lectures now. So now that we've seen this whole idea of which processes are stimulated by insulin and which are stimulated by glucagon, we can actually add an extra level of detail. So we can see that after digestion, we have our glucose. So the glucose can be used once we bring it inside the cell. So to bring glucose inside the cell, we need insulin. So both of these processes that are actually requiring glucose in the cell to be utilized are going to be activated by insulin, because we need to bring the glucose into the cell before we can actually do these processes. So for glycogenesis, we're actually storing our glucose as glycogen, whereas for the glycolysis, we're actually using that glucose to generate pyruvate, which through a series of steps will eventually lead to generation of more ATP. So we see both of these reactions are actually inhibited by glucagon, because glucagon will function to push the glucose into the blood. So if the glucose is in the blood, then it can't actually be utilized by the cell. Also, we saw that the glucagon will actually phosphorylate key enzymes which are required in these processes. So that's another way that the glucagon will inhibit both glycolysis and gluconeogenesis. But that's not all we can do. So sometimes we have the situation where there's actually a low blood glucose level and we need to increase that. So that's when we need to use our glucose making processes. So we can see those two highlighted with the blue names here. So both the gluconeogenesis and the glycogenolysis are glucose making. So the glucose making processes are actually activated by glucagon. So we make the glucose via these processes and then we take it out into the blood. And by pushing that glucose into the blood, we increase the blood glucose level. So both of these processes that are activated by our glucagon can increase the level of blood glucose. So we have these two ways of making glucose. We can either break down glycogen into glucose via the glycogenolysis, or we can actually make the glucose from the non-carbohydrate source of pyruvate via gluconeogenesis. However, both these glucose making processes are actually inhibited by insulin. So insulin will actually promote the usage of glucose for other processes. So it'll actually promote the glucose being used for glycogenesis and also glycolysis instead of being taken out of the cell. And the other thing insulin does to aid this process is it will actually dephosphorylate the key enzymes required for glycogenolysis and gluconeogenesis, which actually slow down and stop these reactions. So we talked about that a few slides back. So if you're not sure of how the role of the phosphorylation allows insulin and glucagon to inhibit these processes that we see here, you can look back at that part of the session. Alright, so we've talked a lot about glucose and how we can control its level in the blood, and we've briefly referred to the pancreas, but let's have a little bit more of a look at the pancreas and its functions. So essentially, the pancreas is the structure we see here in the orange. So it contains important hormones and also enzymes that are involved in digestion. For example, enzymes that can break down dietary protein, lipids, and also carbohydrates. But we're going to focus on its function in releasing hormones. Alright, so the pancreas actually has functions in both releasing hormones into the blood and also releasing enzymes into the small intestine. So for this reason, we call it both an endocrine and an exocrine organ. So the islets of Langerhans, they are the region that will actually release the hormones into the blood. So this will be the glucagon and also the insulin that we've talked a lot about. Whereas the exocrine, the Acini cells, actually release enzymes into the small intestine. So these are our digestive enzymes. So this will allow us to break down our proteins, lipids, and also carbohydrate that we've taken in through the diet. So we can see how these particular hormones and enzymes are actually delivered. So we have the insulin and glucagon, which are in the beta and alpha cells respectively within this pancreatic islet, and we release those hormones via a vein into the blood. So this is how we're going to have the release of the hormones into the blood system, and from there they can have the effect of increasing or decreasing the blood glucose level.